molecular mechanisms can be very complex and overwhelming. All right, so the last class, we said the G protein, of course, is heterotrimeric. All right, that we have the G alpha, beta, and the G gamma subunits, and in the resting phase, these three are closely associated. All right, the G alpha, the G beta, and the G gamma. We said the G alpha and the G gamma, they are anchored on the inner side of the plasma membrane via a lipid anchor. Then the G beta is now kind of sandwiched between them. Okay, so now the G alpha, like I said in the last class, the G alpha is very important because on activation of the G protein coupled receptor, it dissociates from the G beta gamma complex. And when the G alpha dissociates, it goes to activate other cascades of the molecular mechanism. Okay. Now, the G beta gamma complex also play a role. It's not totally dormant or it's not totally idle. It also plays a role. It can also activate some other molecules in the cascade. Okay. So let's take a look at the different types of G alpha that we have. Okay. And this G alpha goes ahead to activate other messengers down the chain. Okay. Now we have the G alpha S. From the name, it's stimulatory. Now, and the primary members are G alpha S and the olfactory G protein. Okay, we perceive smell because of the G proteins, and the type of G protein we use is called the G alpha olfactory, and it's the same mechanism as the G alpha stimulatory. Okay, or the stimulatory G protein. So what happens? An agonist binds to the N terminal. Okay, in the last class, we said that the amino acid has two ends. I said amine group, but technically in chemistry, in chemistry or biochemistry, I think we should have used amino group. Okay, but they are almost the same thing. In fact, they are synonymous, but the impeccable thing to say is that we have the amino group end and the carboxylic group end. So our ligand binds, or our agonist binds to the N terminal in the extracellular space. Okay, when it binds to the N terminal in the extracellular place, there's a conformational change that happens to your G protein coupled receptor. Now, this conformational change now causes stimulates the G alpha. Then the G alpha has like a guanine exchanging factor, okay, that exchanges GDP for GTP. Now, and a dissociation takes place. So the G alpha dissociates from the G beta G gamma complex. So what happens in the case of G alpha S? Okay, so take for instance, if we have hormone or neurotransmitter bind, for example, epinephrine glucagon, bind to the G protein coupled receptor, particularly the one coupled with the G alpha stimulatory. Okay, as a conformational change, I already said this. Then GDP is exchanged for GTP. Then this the G alpha has GTP now dissociates from the beta gamma subunit. Now this G alpha has GTP now goes ahead to activate adenylate cyclase. Now, in biochemistry, there are different enzymes. Now, when we say cyclase means that it's going to convert the molecule from a linear structure to a cyclic structure. That's what that means. So, when we say G alpha S GTP is activating adenylate cyclase, then that means that it's going to convert a molecule from the linear structure into the cyclic structure. Okay, so now this adenylyl cyclase, what does it do? When the GS alpha or the G alpha S GTP activates the adenylate cyclase, it converts adenosine triphosphate into cyclic adenosine monophosphate. Okay, so it converts adenosine triphosphate into cyclic adenosine monophosphate. Okay, so like we said, anytime you hear cyclase, it's just to convert a linear molecule into the cyclic form. Now, so it is this CAMP that now acts as a second messenger to propagate the signal inside the cell. So what does CAMP do? CAMP activates protein kinase A, and when CAMP binds to, there are units of the protein kinase. We have the regulatory subunit and we have the catalytic subunit. When CAMP binds to the regulatory subunit of protein kinase A, it makes them to release the catalytic subunit. Then, okay, I should also say this. When we say kinase in biochemistry, what a kinase does is to add phosphate groups to molecules. So kinase phosphorylates, all right? Cyclase convert a structure from the linear form into the cyclic form, while phosphatases removes dephosphorylates, okay? Now, in biochemistry, let me repeat that. Kinases phosphorylate, while phosphatases dephosphorylates, okay, or dephosphorylate. Then cyclases, we convert a molecule from the linear form into the cyclic form. So when the catalytic subunits of the protein kinase A are released, okay, now this catalytic subunit then goes on, because it's a kinase, then it goes on to now phosphorylate target proteins, okay. So what are some of the target proteins that protein kinase A phosphorylates? We have transcription factors like CREB and CREB-CAMP. CREB is CAMP response element binding protein. When it phosphorylates CREB, CREB can then go on to elicit pharmacologic effect. Then it can phosphorylate ion channel, can phosphorylate metabolic enzymes. And once this take place, then we can have our cellular response, okay? Now, so what happens? Let me summarize that again. For the G-alpha S, okay, for the G-alpha S, what we have is that once the receptor is activated, the G-alpha S exchanges GDP for GTP. Now, this G-alpha S GTP complex then dissociates. Now, the G-alpha S GTP complex then goes to activate adenylate cyclase. Now, and these adenylate cyclase, like we said, we convert ATP into CAMP. Then CAMP, we in turn go to activate our protein kinase A, like we said. It binds to the regulatory unit and release the catalytic subunit. Then we said kinases generally phosphorylate. Then the kinases, we now go to phosphorylate our effector proteins, okay? So, and some of these can be CREB, it can be ion channels, it can be metabolic enzymes, as the case may be. Then we can then have a cellular response. So what happens in the heart? In the heart, this signaling opens calcium channels in the heart, and as a result, we can have strong contraction. In smooth muscle, okay, PKA lowers calcium ion availability. So in smooth muscle, we have relaxation, not contraction. Okay, so in the heart, we have stronger contraction. Okay, of course, because what we have there, we have myosin, cardiac muscles, which are quite different from smooth muscles. But in the smooth muscles, we have relaxation. Okay, so that's why beta agonists, albuterol, that you popularly know as salbutamol, can relax the bronchi smooth muscle. Okay, it's via this G-alpha S cascade or mechanism that it does that. Then in the glands, the G-alpha stimulatory can boost secretion. Okay, in the nucleus, it activates CREB, and this CREB can alter gene expression. All right. Now then, protein kinase A also can activate metabolic enzymes. Then it inserts aquaporins into the membrane. This is of importance when we are talking about water retention and the conservation of osmolality and all of that. Then the protein kinase A also activates protein pumps. Okay, so that means if there is an agonist of G-alpha S, it can cause hyperacidity. All right. So how will this signal be terminated? All right. So how do we terminate this signal? Okay, so the termination is by, there's an intrinsic, you see, your G-alpha has an intrinsic phosphatase activity. And you know, I told you that kinases phosphorylate, while phosphatases dephosphorylate. Okay, so our G-alpha has an intrinsic phosphatase activity. So what happens is that the phosphatase activity, or the intrinsic phosphatase activity, then convert GTP into dephosphorylate GTP. So we now have our G-alpha GDP. This now goes back to reassociate with the G-beta gamma complex, okay, and makes the receptor return to its resting phase or the inactive phase. Then also, you know, because we have we have CAMP as a result of adenylate cyclase, okay, phosphodiesterase also can degrade CAMP and convert CAMP back into adenosine monophosphate. and this phosphodiesterase also reduces protein kinase A activity. So phosphodiesterase also help in terminating the signals. Okay. Then another thing we can have is what we call desensitization. All right, we can have desensitization when a receptor is exposed to a ligand for a very long period of time, then desensitization can occur. Now, the desensitization is going to be by a phosphorylation of the G protein coupled receptor itself. Now, and this phosphorylation, like we said, when we say anytime we say phosphorylation, is by kinases. Okay, so this phosphorylation is going to be carried out by G protein coupled receptor kinases. Okay, so this GRKs phosphorylates the G protein coupled receptor itself, and after the phosphorylation, all right, this alters the shape and the conformation of the receptor. Okay, so when the GRKs phosphorylate the receptor, of course, fundamentally there's not a change in the chemical structure of your receptor, and as a result of that, it will alter its function. Okay, now then after this phosphorylation, arrestins now bind to this phosphorylated receptor, which will now block further G protein activation. Then your G protein can now be, after phosphorylation, can be internalized by your cell, and this is endocytosis, and it can be digested. Okay, that way. So this mechanism, all right, is the mechanism behind desensitization when you are exposed to an agonist for a very long period of time. Now, there are typical examples that I can give you. Take, for instance, for someone who is taking beta-2 agonist, all right, like albuterol or salbutamol, what you realize is that for bronchodilation, maybe an asthmatic patient, what you realize is that on prolonged use, the effect of this beta-2 agonists begin to reduce. Okay, so the mechanism behind this is the phosphorylation of the G protein coupled receptor. The G protein coupled receptor becomes phosphorylated, and once it's phosphorylated, arrestin binds, and when arrestin binds to the phosphorylated receptor, this can block further activation. All right, then there can be internalization of the receptor via endocytosis. All right, and when this happens, a receptor reduces, then there is desensitization. Okay, so you do not necessarily have to tell me the effect, but who can tell me the cascade of events that happens in a G-alpha S coupled receptor? You can tell me the cascade of events that happens in a G-alpha S coupled, in G-alpha stimulatory coupled receptor. Anybody? Anybody? We have said it too. There is adenylyl cyclase and the likes like that. So who can tell us the cascade of events? Okay, let me repeat it again now. I said for the G-alpha stimulatory, okay, if your G protein coupled receptor is G-alpha stimulatory, our concern in the heterotrimer is the G-alpha. Now, it's not that the G-beta do not have their roles. Okay, the G-beta 2 can activate some pathways, but our major concern is the G-alpha. So the subtypes of the G protein coupled receptors that we'll be looking at, the subtypes is basically referring to the G-alpha. So the first one we have taken a look at is the G-alpha S, which is the stimulatory. And the mechanism is that once an agonist binds to the amino terminal or to the N terminal of your receptor, okay, it activates that receptor. There's a conformational change in the receptor, and this conformational change stimulates the G-alpha S to exchange GDP for GTP, and once we have G-alpha S GTP, there's a dissociation. Okay, it dissociates from the heterotrimer. Now then, this dissociation, the G-alpha S GTP then goes to activate adenylyl cyclase, and I told you adenylyl cyclase will now convert ATP into cyclic adenosine monophosphate. Now this cyclic adenosine monophosphate will in turn go and bind to the regulatory unit of the protein kinase A and release the catalytic subunit of the protein kinase A. Now, once the catalytic subunit of the protein kinase A is released, and you know that, like I said, kinases phosphorylate. This protein kinase A then goes to phosphorylate either your ion channel, your metabolic enzymes, and the likes, and then there is now cellular response. Okay, cellular response, and we saw some of the effects, the organ effects of these different responses. Then we discussed mechanisms of termination of the activity of these G-alpha S or the G protein generally. Talked about phosphodiesterases, the intrinsic phosphatase activity, and desensitization. The same thing happens in opioid. When you are taking opioid for a very long period of time, then there is desensitization, and this can lead to tolerance. So this desensitization mechanism, receptors desensitization, is one of the mechanisms of tolerance amongst many others. Okay, tolerance can also be due to enzyme inhibition. No, enzyme induction rather. I beg your pardon. Enzyme induction. But I'm still going to take you enzymes. So when we get to that, we'll talk more on the mechanisms of enzyme induction for tolerance, all right, and withdrawal syndrome. Okay. So in the heart, we said calcium entry increases. Then we have stronger and faster heartbeats. Then there is contraction of the cardiac muscles. But for the smooth muscle, we have relaxation. Then we said it activates metabolic enzymes. So there will be increased glucose and fatty acid release. In the kidney, there will be water retention rather because of the insertion of aquaporins into the membrane. Okay. Then in the stomach, of course, there will be increased acid secretion, and this is by the histamine 2 receptors. Okay, so let's see some of the G protein coupled receptors that employ the G-alpha S mechanism. Okay. Now, typical examples include our beta-1 adrenergic receptor. It employs the G-alpha S subunit. So, because of this pathway, then we are going to have increased heart rate and contractility. And this is the mechanism by which adrenaline contracts the heart and increases the heart rate. Also, some of its agonists, like dobutamine, via the beta-1 and the G. Okay, so beta-1 is the receptor. It's coupled with the G-alpha S. Okay. Then for the beta-2 adrenergic receptor, the beta-2 adrenergic receptor is usually found on smooth muscles. So, like we said, that the G-alpha S contracts cardiac muscles. Okay, while the G-alpha S relaxes smooth muscle. So we are expecting contractility in the cardiac muscle, while in the smooth muscle, we are expecting relaxation. So activation of beta-2 receptor, which is coupled to G-alpha S, relaxes the smooth muscle, particularly the bronchi smooth muscle and the uterine smooth muscle. Of course, you know that we have smooth muscles in the uterus, then we have smooth muscles in the airways. The middle layer of the uterus has the myometrium, and the myometrium is a smooth muscle. In fact, that is why sometimes in cases of threatened abortion, you can prescribe salbutamol, because salbutamol relaxes the smooth muscle via the beta-2 coupled with the G-alpha S. It relaxes the smooth muscle. So salbutamol is one of the drugs you can prescribe to a pregnant mother who doesn't want to lose the pregnancy anyways and is having threatened abortion. Okay, and of course we also prescribe the beta-2 agonist for bronchodilation, or for bronchospasm rather. All right. So now the beta-2 agonist, we have the short acting, long acting. I believe you know all of that. Have you guys done asthma? Have you guys done asthma? I need a response. I need to know that you guys are here.
Yes, sir. Yes, sir.
Okay. So you see now, if I now say, explain the therapeutic role of the G protein coupled receptor in bronchospasm. Of course, you should know that I'm talking about beta-2 adrenergic receptor. I explain the mechanism of anything related to bronchospasm. Then you should know I'm talking about the beta-2 adrenergic receptor. Then you must tell me about, because the course is all about molecular mechanism, you must tell me it's a G protein coupled receptor. Particularly to the G-alpha system is the G-alpha stimulatory, okay? And the G-alpha stimulatory, you talk about the cascade of events. Then you now in turn say in the bronchial smooth muscle, it relaxes the protein kinase A, okay? Relaxes the bronchial smooth muscle. So, and that's the mechanism. Also in uterine contraction, that's also what we have. Then for beta-3, because we said that the protein kinase A also activates metabolic enzymes. Okay, so that means we're expecting the breakdown of complex molecules. Okay, we're expecting catabolism generally, not anabolism. So there will be increased lipolysis in fat cells. Okay, so these we have to, lipolysis means breakdown of lipid in literal term. Okay, so now for the D1 dopamine receptors, it dilates renal blood vessels. The beta-3 adrenergic receptor, of course, will relax bladder, because even the bladder also contains smooth muscle. Okay, so you're expecting bladder relaxation. Then we said that for the G-alpha stimulatory, what it does is that it opens the proton pump, or it enhances the proton pump. So, of course, we're expecting increase in gastric acid secretion. Okay, So then all these other effects, they are just as a result of all the things you have said earlier on. That okay, also the GFR stimulatory also can make the glands secrete their hormones. So this can raise thyroid hormone synthesis, LH and FSH synthesis and all of that. Then it can release glucagon too, and you know what glucagon does? It increases blood glucose generally, because also the metabolic enzymes will be upregulated and all of that. Any question? Do you have any question? Do you have any question? Before I move to the the second one that we refer to as the G-alpha IO, which is the inhibitory. The G-alpha S is the stimulatory, then the G-alpha IO is the inhibitory. Okay, just like in this case of the G-alpha S, that we have the G-alpha S and the G-alpha olfactory, which are the primary members of the G-alpha S. For the G-alpha high and O, the primary members are the G-alpha 1, 2, 3, G-alpha high 1, 2, 3. Okay, then the G-alpha O itself, and we have G-alpha T, which is this is not, there is no G. It's sorry, this should be a mistake. This should be transducin. It's going to be autocorrect like that. Transducin involved in vision. So let's see what happens in G-alpha high O. Okay, we start from ligand binding. That one is common to them all. Now an agonist is going to bind, then activate the G-alpha high O. Okay, so what happens when the G-alpha high O is activated? This one is common to all of them. So who can tell us what happens next if we have an agonist bind to the N terminal? The receptor becomes activated, then changes conformation. Then this will now go ahead to activate the G-alpha high O, which will exchange GDP for GTP. Once we now have G-alpha GTP, it dissociates from the G-beta gamma complex. Okay, once it dissociates, then we can now talk about what it is going to now activate. Okay, so what happens in the case of after dissociation, the G-alpha IO GTP will now bind to adenylate cyclase, but in this case, it is not activating adenylate cyclase. It is going to inhibit adenylate cyclase. Of course, you know that the activation of adenylate cyclase is going to cause the conversion of adenosine triphosphate into cyclic AMP. But in the case of the G-alpha IO, we are not going to have this, because the G-alpha IO is going to inhibit the adenylate cyclase, and once it inhibits it, then it shuts it down. Okay, the shutdown is just for the lay person. Okay, so but of course it inhibits, it binds to the adenylate cyclase and inhibits it. So CAMP levels drop, and once CAMP level drops, there will be a reduction in the activity of protein kinase A. All right, then we will now have all those cellular responses. So what happens once there is a reduction in CAMP and there is also a reduction in protein kinase A activity? Then many phosphorylation will not occur, and if there are no phosphorylations, then we do not expect to see phosphorylation of ion channels. Metabolic enzymes will be affected. Transcription factors like CREB, transcription factor like CREB will also be affected. So generally, the cells just becomes less active. Now, like I told you that the beta gamma complex are not totally useless. They also interact with the cascade in a way. So for the beta gamma subunit, okay, so they can open a G protein associated potassium channel. We call it GIRK. And once they open this, there is a papularization. We talked about polarization, depolarization, repolarization, papularization under anticonvulsants. Then they can they will also close the voltage gated calcium channel. Then there will be less calcium entry. They reduce neurotransmitter release in the neurons. Okay, so generally what the G-alpha IO does is just to quieten the cells. So let's see what this effect will be like for cell response. Okay, now for the heart, when we have activation of the G-alpha IO, it reduces the heart rate, unlike the G-alpha S. Okay, it reduces the heart rate. What we will begin to imagine are the direct opposites of what we have in G-alpha stimulatory. Okay, because they interact with the same mechanism. which is adenylate cyclase. Okay, so we begin to now imagine what happens. Just anything we have said under G-alpha stimulatory, just begin to reverse it. Okay, so for the heart, we are going to have a reduction in the heart rate. This is the action of the muscarinic 2 receptor. All right, because there is less calm, fewer opening of calcium channels, and all of that. So, we also said that the G-alpha stimulatory ensures neurotransmitter release, but there will be a reduction in neurotransmitter release from the neurons, okay? Then because of less calcium entries also, there will be a reduction in insulin release, okay? And this is mediated via the alpha-2 receptors. Then for lipolysis, we said that the G-alpha stimulatory activates metabolic enzymes, but the G-alpha inhibitor is going to slow lipolysis, and this is because the enzymes need activation by protein kinase A, so there will be slowing of lipolysis. Now, in G-alpha stimulatory, we have relaxation of the smooth muscle, but in G-alpha inhibitory, we are going to have contraction of the smooth muscle. So let's go. How is this signal going to be terminated? It's the same way as in G-alpha stimulatory. There's an intrinsic activity of the G protein. That intrinsic activity is a phosphatase activity. And like we said, that phosphatase is going to remove phosphate. And once it dephosphorylates, GTP becomes converted into GDP. And this can terminate this signaling, okay? Then phosphodiesterase 2 in this case too can help to terminate this signaling. Then, like we also said, our desensitization, when you keep using the drug, there can be desensitization, and desensitization is via two things: the GPCR kinases, which phosphorylates the receptor, and arrestin binds. After arrestin binds, there can be internalization of our receptor via endocytosis and all. So let's see some of the organ effects. Like play, we are going to finish the protein coupled receptor, then move into ion channel. Okay. Now there's still a categorization I'm going to give you, but I want us to discuss all the molecular mechanisms before I now tell you the classical receptors, the ones that are non-receptors, and all of that. Okay. So now let's see some of the effects, the organ effect or systemic effects of the G-alpha inhibitory. We said for the G-alpha stimulatory, there's increase in heart rate, but here there's a slower heart rate, okay, because of the effect on the natural pacemaker, the sinoatrial node and the atrioventricular node, okay, of the heart. All right. All these why we already know that. Then there's less firing, less neurotransmitter release. In the pancreas, there is low insulin release, okay. Then there's slow lipolysis. For the smooth muscle, there's contraction, unlike in the G-alpha stimulatory where there is relaxation. Okay? So let's see some of the classical or typical examples of these receptors. Okay, so I just have 1 hour because I know you guys are busy. Akaps, I don't know if it's going to be possible to have a constant class. If it's not possible, then no problem. I'll keep reaching out to Akaps to just get an hour per week. An hour per week is what I need to wrap up my part with you guys. I'm not taking you 501. I'm basically in charge of 503. Okay, I don't know if there's a 501, but I'm not part of that lecture. I'm part of the 503 lecture. Okay, so, and I just need an hour per week. I don't think we have to increase it. To run through everything I need to run through with you guys, then we are home and dry. So if it can be constant, no problem. But if it's not constant, then there's also no problem. We will still do what we want to do. Then we can have enough time for revision. But I do not want any week to go by without we meeting, so that there can be our revision class. We can discuss some practice questions. Then if there are gray areas that you need more clarification on, you can ask me. Then we discuss the practice questions. And it's a win-win for both of us, for the lecturer and the student alike. I'm able to finish my allocated topics. Then you have enough time to then read through the materials, and we have enough time to treat some questions and all of that. Because, of course, when I treat questions with you guys, they are very important. But any which way, there's a part for it's more like a 50-50 win, like I said. There will still be the back-breaking lecturers' questions, which will be 50%. Then there will be 50% too for the students, if not more. Okay. So now, the alpha-2 adrenergic receptor uses this G-alpha HIHO. Okay, so of course we are expecting things like low sympathetic outflow, sedation, low BP, because adenylyl cyclase is inhibited. Of course, you know that. Then the M2 receptor 2, okay, slows heart rate. All right. The dopamine 2 receptor, we now inhibit prolactin and release, and this is how bromocriptine works. Okay, bromocriptine tones down prolactin. All right, because it binds to the dopamine 2 receptor, whose mechanism is via the G alpha IO. Okay, so then prolactin goes down. Some people, you know the implication now when prolactin goes down. Okay, so what are some of the implications? Number one is that when there's hyperprolactinemia, there can be infertility, because when prolactin is high in the bloodstream and then there is infertility, there might be, it's prolactin itself has contraceptive effects, kind of. Okay, so that's why you see that when the mother is lactating, particularly early post-delivery, if the mother has unprotected sexual intercourse, there's a low likelihood that in fact she may not get pregnant. But of course, it's not 100%. It's not even a good contraceptive method. But physiologically, when prolactin is flowing in your body, it inhibits conception. Okay, so bromocriptine can be used for hyperprolactinemia. Then opioid receptors too, the mu kappa and the mu kappa and the deltoid, they also have this mechanism of G alpha IO. Okay, so and this causes analgesia, sedation, reduced neurotransmission. GABA B receptor, also baclofen. Now, you know, we said the cell, in a way, there's a reduction in the activity of the cell. So technically, anything that is going to probably calm you down, let me say it in that way, may be working via the inhibitory mechanism. Okay, so baclofen too works via this mechanism. It reduces calcium entry. All right, so yeah, baclofen also generally relaxes the muscle, reduces firing and all of that. Okay, so there are also other receptors that work via this mechanism, like the adenosine receptor. The adenosine receptor slows AV conduction and all of that. Okay, so let's go to, oops, we have just 8 minutes. And I want to show you how to prepare your protein, so that you guys can go ahead and prepare your protein. Okay, so please, let me, permit me to crave your indulgence. Let me use the next 5 minutes to move into molecular docking, so I can show you guys how to prepare your protein. Which group should I use? Which group? Let me see. Epilepsy. Which group should I use? I'll just quickly show you guys how to prepare your protein using the Biovia Discovery Studio. Then I'm going to encourage you to go ahead and install the other applications we have. Which group? Group 2. Group 2, okay. Okay, so let me bring down this. Stop sharing. Then I think I should share my entire screen. But before I do that, let me open Discovery Studio, so that. So sorry, under 10 minutes, I will just teach you the next thing. Then we can go ahead. Please, not all groups should choose alkaloids. I don't think we should all use alkaloids. We can use other classes. So, but I'm still going to look into that. But not all groups should use alkaloids. No, we can. Group 2. Oops, no. Group 2 is hypertension. Then your target is 1086. Okay. 1086. I think I have this protein. 1086, but any which way. Please, just a moment. Let me bring up the protein. 1086. Yes, I have the protein. Okay, so now I think I can share my entire screen with you guys. This is not going to take more than 10 minutes. Should I share my entire screen? Yeah, let me share my entire screen. So can you guys see my Discovery Studio? Can you guys see my Discovery Studio? Hello, can you guys see my Discovery Studio?
Yes, sir.
Okay, so you would realize that in this, we have so many things here, all right? The red tiny dots are water molecules. There are so many things that we have here. So anyways, the first thing first is to, if you can reduce the size using the ball of your mouse, okay? If you have, I think it's only possible if your mouse has a ball, okay? So you if you take down the ball, then you reduce the size of this molecule in the Behavioral Discovery Studio interface. Then if you keep turning up the ball, then you increase the size and all of that. So the first thing first is to check the interaction. Okay, now because I clicked to the interaction, then it's not showing me anything relevant. All right, but the first thing first is here. There is a defined ligand. Okay, so you just look for where your ligand is. I think in your own case, your ligand is lisinopril. So it's here. I already selected it. So selecting it, if I say show 2D, okay, still not showing me. So that means I have to define it. Let me say define ligand, okay? So I think we are ready. We are good to go. Then show 2D. Yes. Now, this is now showing me something interesting. So this is what you do. Once to prepare your protein, you need this image. The reason why you need this image is because there are bridging waters. If you check, there are water molecules interacting with your ligand, okay? Now, these water molecules, ideally, in the past, the way we dock is that you remove all water molecules. You remove your ligand, then we only have the amino acid chains of the protein. But sometimes it is important you keep your bridging waters, particularly the water that the ligand is in, the waters, I can use the plural, this is chemistry. Now, the waters the ligand is interacting with. The reason is that if you remove the waters the ligand is interacting with, there will be a little conformational change in the 3D pose of your ligand. So sometimes in docking, we just retain the water molecules that our ligand is binding with. In this case, we have HOH2393. Anywhere you see HOH, that's a water molecule. HOH2392, HOH2409. So now, the next thing you do is just that you have to note it down. For me, I just, when I screen grab this, I now write out all the water molecules that is interacting with my ligand. In this case, these ones, HOH2029, HOH2365, you can delete them. But if you want to repeat this same conformation, then you can keep these ones, because in a way, although it's seeming that there is no interaction, but the aromatic ring is almost being guided by these water molecules. But because there is no direct interaction as such, you can remove these water molecules, okay? So, but the ones that have dash interaction, you can retain the water molecules. In fact, if we remove these ones that are close to the aromatic ring, we still have a little change in the pose of your ligand, because it appears that they are useless, but they still have their effects, okay? So, but you can remove all these ones and just retain the ones that are directly interacting with your ligand. But do not delete any amino acids. The amino acids are part of the protein and they are very important. So you note down the water molecules that are directly interacting with your ligand. Now, another thing is, once you have noted that down, now it's very easy. You can delete other things. Now, another thing I'm seeing here for this group is that we call some enzymes metalloenzyme. That means these enzymes have metals attached to them. And I think an example is angiotensin converting. This is angiotensin converting enzyme group 2, right? Group 2. Hey. Am I the only one in this class? Let me go back. Group 2, this is angiotensin converting enzyme. okay, if you are not responding, no problem. But it's a metalloenzyme, if that's what they call them, if I can still remember very well. So you can see that there is a zinc molecule here. Your ligand is interacting with zinc here, okay? So you still have to retain that zinc. You still have to retain that zinc. Okay, now let me tell you why the things you have to retain in your protein molecule are important. Number one is because now we will remove this ligand. This ligand is complex to the protein molecule, but we are going to remove it, okay? And when we remove it, we will redock it back into the protein molecule. Then it must have a similar pose as this to validate your docking, okay? This is important for docking validation. It must have a similar pose as this for your docking to be validated, okay? Or else, when we redock and we check and it doesn't have a similar pose, journals will not accept your publication. We call it docking validation, okay? Although sometimes they say the docking validation is still elementary, that you need molecular dynamics and all of that, but I'm going to explain all of that to you. But I have revised papers in...
Yes, sir.
There's a question? Is there a question? Okay, so let me continue. So when we do the redocking, the first thing we are going to do is redocking, guys. And when you redock, it must have a similar pose as this and similar interactions, okay? That's when we can say that your docking procedure or your docking protocol is accurate, okay? So now so this is what you guys are going to do next, all right? You are going to prepare this protein. You remove all the water molecules that are not interacting with your ligand. You already have this picture. So just remove all the water molecules, okay? Now, the chain A, the first one here contains my amino acid. The next one here contains my ligand and some other supporting structure like zinc. And zinc is here. Zinc is also interacting with your... see, as you click, if this molecule is here, you will see it here. So if I click lisinopril, you can see that lisinopril is highlighted. Everything is highlighted here. If I click zinc, zinc is highlighted, yes. Zinc is interacting with carbonyl group here, okay? So zinc is highlighted here. If I click chloride, chloride is not interacting, okay? So if I click this chloride too, there is no chloride highlighted here. So that is telling you that you can delete this chloride. And the way you delete is that you just click and press delete on your keyboard. I've deleted it. You click and press delete. I've deleted it. It's the same thing you do in your Microsoft Word. If you have deleted in error, just press Ctrl Z. And see, one chlorine molecule has jumped back. Then you press another Ctrl Z. Oops. No, sir, the two chlorine molecules are back now. So to delete anything you don't want in your protein, you just click it and press delete. It will be deleted. Now, like I told you, I said we need to remove this ligand, this lisinopril, LPR702, and keep it. The reason why we need to do that is that we have to redock it, okay, into this protein and see if it's going to give us a similar pose. And that's what we call docking validation. All right, are you guys following? Now, how do we remove it? It's the same way. Whatever you do on your Microsoft Word, that's why Discovery Studio Visualizer is very simple and user friendly. So I will click on the lisinopril, then Ctrl X. Ctrl X means cut. Now the ligand has disappeared. Okay, I've removed it that way. Then you come to your file, new, open a new molecule window. Now this is a new molecule window. Then press Ctrl V. Now here you have your lisinopril. Okay, I've removed this lisinopril. Now let me now save it. I will now save the lisinopril. I'll say save as lisinopril. Let me save it under my downloads. Let me say native ligand. We call it the native ligand. Let me say native underscore ligand, or you can give it the name lisinopril. Okay. Now the reason why you have to remove the native ligand and redock is because this native ligand, as you are looking at it now, it has 3D coordinates. Okay. Now, if you go and download fresh lisinopril from PubChem, it will not work for your docking validation. All right. Now, because this lisinopril itself has what we call 3D dimensions, okay, which is important for the redocking validation. Some of this concept might be too complex, but it's simple. You guys will understand it. Now, see, this is my native ligand. If I right click and say, that's the lisinopria I removed from the protein. If I right click and say open with, okay, if I say edit in Notepad, you will see what my Notepad is going to open. Oops. The file is corrupt. The file is corrupt. Oh, sorry. I think I caused the problem. Let me save again. Save as. Yeah, I'm supposed to save this as MDLMO SD. You pick MDLMO SD files. That's how we are going to save it. Okay. Then native_ligand. Then save. Okay. I can then go back, download, document, download. I need to know which is which. I think I should delete this. Then is this now? If you right click this and open and say edit in Notepad, yeah, this is it. So you realize that this is your lisinopria. Okay. Now, all the chemical structures you see on PubChem, they're actually coded in binary language. Okay, you know what the language the computer understands is binary, 1010101010. So they have been coded in binary language and all of that. Now then we now have coordinates. This 43.3 something you are seeing here is the x coordinate. This is your y coordinate. This is your z coordinate. If I alter this in the Notepad, then your molecule will be deformed. So most of the time, we don't go into this Notepad to check your molecule because you can end up deforming your molecule, because once you alter anything, it changes the string of the molecule. Okay, so the reason why it's important you remove that native lisinoprile and not download a fresh lisinoprile for your redocking is because if you download a fresh lisinoprile, it's going to have different 3D coordinates. And these different 3D coordinates, it might be difficult for you to get the same pose that we got from protein data bank rather. Okay, all right. So now I have removed the ligand. So now let's assume that there is no water molecule interacting with our ligand. I've removed ligand from this protein. So let's assume there is no water molecule interacting with this ligand. The third A consists of all the water molecules, HOH, HOH, HOH. I will just come here. If there is no water molecule interacting, you see that on clicking, it highlighted all the water molecules. I will just press delete. Then all the water molecules are gone. Then also here, we have chlorine. I've checked my 2D interaction. Chlorine is not interacting. I delete. Then I delete. In my 2D interaction, zinc is interacting. So I will leave zinc. But this is not interacting. Then I'll delete. Okay. Once I've done this, I have prepared my protein. This is my protein alone with the important things, okay, that I need. Of course, you know that when I checked this interaction, some water molecules were interacting. But I can't jot down now and begin to delete the ones that are not interacting and retain the ones that are interacting. But you can do that, okay? When you note the water molecules that are interacting with your ligands, then you selectively delete the water molecules. So once you are done cleaning your protein like this, then you come to file and do save as, okay? Don't save as Discovery Studio files. You don't save as that. For the protein, you save as PDB, protein data bank files. That's how you save your clean protein, okay? Then your ligand, you save it as mole SD files, okay? Mole SD files. You save your protein as protein data bank files. So I'll just click this. Then I can say 1086_cleaned. And you just click save. Once you save it like that, you have saved your cleaned protein target. Then we can then now move on to the next thing. So this brings us to the end of what we have to discuss today. So any question so far from the few things I've demonstrated? Any question? Any question? Okay, all right. If there are no questions, we meet next class because it appears that everybody has left. Only a few people stayed back. But if there are questions in the course of the week, maybe you tried cleaning your protein and all of that and you didn't get it, you can reach out to me. I'll be ready to explain to you guys. But I will also send the videos to you guys almost immediately so that I can guide you when you are trying to clean your protein. Okay? So questions. If there are no questions, all right, we meet next week again. Thank you, guys.
Thank you, sir.
You're welcome.